Structure of a leaf. As we have learnt, plants make their own food in a process known as photosynthesis. The process of photosynthesis occurs mainly in the leaves of plants. Therefore, to understand the process of photosynthesis, it is necessary to study the external and internal structure of a leaf. External structure of a leaf. A leaf is a thin, flattened organ which is attached to the stem or branch of a green plant. A typical simple leaf consists of a flattened surface called lamina. The lamina is green in color and contains the photosynthetic tissue. In dicotyledonous plants, simple leaves have a thick midrib which runs in the middle. From the midrib arise small veins that run through the lamina, forming an extensive network of veins. In monocotyledonous plants, the midrib is absent, but small veins run parallel to each other. In some plants, the leaf is attached to the stem or branch by a petiole, while in others the leaf is attached directly to the stem. In most monocotyledons, leaves are attached to the stem by a modified petiole, the leaf sheath. Internal structure of a leaf. When observed under a light microscope, a transverse structure reveals the internal structure of a leaf. A leaf has several parts which have different roles that they play in the functioning of a leaf. These parts are: cuticle. This is a thin non-cellular, waxy, waterproof, transparent layer that covers the surface of the leaf. It reduces excessive loss of water and protects the inner tissues of the leaf from mechanical damage. It also prevents entry of disease-causing microorganisms. The cuticle is transparent to allow light to penetrate through it into the photosynthetic cells. Epidermis. This is a thin, transparent tissue, usually 1 cell thick, on the surface of the leaf. It allows light to penetrate into the photosynthetic cells. The epidermal cells have no chloroplasts except the guard cells. Chloroplasts are the organelles that contain the photosynthetic material known as chlorophyll. The epidermis secretes the cuticle and also protects the inner tissues from mechanical damage and entry of disease-causing microorganisms. Guard cells. These are special bean-shaped epidermal cells, mostly found on the lower surface of the leaf. Unlike the adjacent epidermal cells, guard cells contain chloroplasts and are able to carry out photosynthesis. They control the opening and closing of the stoma. They also control water loss. Through the stomata, air moves in and out of the leaf. Palisade layer. This is a layer of cells located beneath the upper epidermis. It consists of cylindrical cells, closely packed together with the long axis perpendicular to the surface. They have numerous chloroplasts containing chlorophyll, which is necessary for photosynthesis. Their position and arrangement enable them to receive maximum sunlight. Spongy mesophyll layer. This is a layer of cells between the palisade and the lower epidermis. The cells are irregularly shaped and loosely arranged, creating large air spaces in between them. The air spaces provide communication pathways through which gases diffuse between the cells. Unlike the palisade cells, these cells contain fewer chloroplasts. This explains why the lower surface of the leaf is lighter in color than the upper surface. Leaf veins. The leaf has an extensive system of veins which ensures that every cell is close to the supply of materials. Each vein is a vascular bundle which consists of xylem and phloem tissues. Through the xylem, water and mineral salts are conducted from the roots to the cells of the leaf. Manufactured food is translocated through the phloem from the leaf cells to the rest of the plant. Adaptation of the leaf to its photosynthetic function. Leaves possess special structural features which adapt them to carry out the process of photosynthesis efficiently. These structural features include the following: a broad flat lamina which provides a large surface area for the absorption of carbon dioxide and sunlight. The thinness of the leaf lamina allows light and carbon dioxide to pass through a short distance to reach the photosynthetic cells. The presence of stomata ensures efficient diffusion of carbon dioxide into the leaf. The cuticle and epidermis are transparent to allow penetration of light to the palisade cells. The palisade cells contain large numbers of chloroplasts, and their arrangement and location next to the upper epidermis enables them to receive maximum sunlight. The existence of extensive veins that contain vascular bundles which conduct water and mineral salts to the photosynthetic cells, and removes the products of photosynthesis. Spongy mesophyll cells are loosely arranged, leaving large intercellular air spaces that are interconnected for rapid diffusion of carbon dioxide into the cells and oxygen out into the atmosphere. The regular arrangement of leaves on the stem minimizes overlapping and overshadowing, which ensures that all leaves receive light. This type of leaf arrangement is known as leaf mosaic. The chloroplasts. These are disc-shaped organelles found in the cytoplasm of some plant cells. Each chloroplast is bounded by double-unit membrane with an outer and inner membrane. Internally, the chloroplast consists of layers of membranes known as lamellae suspended in an aqueous matrix called stroma. The lamellae may at certain intervals form several layers of membranes grouped together to form a granum, plural, grana. Chlorophyll molecules and other photosynthetic pigments are located in the granum, which provides a large surface area to accommodate a large number of chlorophyll molecules. Chlorophyll molecules absorb light energy which is necessary for photosynthesis to take place. The stroma contains enzymes that speed up the process of photosynthesis. Thank you for watching this biology lesson. Please subscribe to this channel and other Genius channels relevant to your level of study.